Cek, halo, halo, cek, cek. Kalau mic yang di meja sana, ini sudah. Mana mic di meja? Cek, halo, 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 cek. Coba kau dengar dulu, masuk. Cek, cek, halo, 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 masuk. Sudah, sudah suaranya. Tidak tertangkap. Cek, cek, halo, tidak tertangkap. Mas, cek, halo, halo, cek. Cek, ini masuk. Ini masuk. Cek, halo, halo. Iyo, ada dua sumber mic-nya. Ini, Pak. Ini, ini bagus ya. Ini kan yang turun dia. Cek. Halo, halo, halo. Sementara itu kita pakai, bro, mas bro. Ini kan cek masuk. Halo, naik. Supaya satu ini saja dipakai. Makanya dia dual terjadi.
Ya, satu
Satu
tidak
tas
tidak connect ke
satu
Zoom.
telepon ke Zoom.
Oh,
Itu yang menjadi masalah, suaranya kecil.
oh begitu.
Jadi sehingga dia double apa? Double keluar.
Oh, jadi tutup saja, matikan saja.
Iya, makanya kita tidak. Cik, cik. Ah, ini yang masuk, Pak.
Halo, cek. Tuannya untuk bisa
Cek, cek, satu, cek. Halo. Halo, halo, cek, 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 satu.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And good afternoon, everyone. Thank you very much for still stay with us. And I'm the session chair for the third plenary session. So this is the last plenary session for today's conference. So here we have two keynote speakers for our plenary session. The first one is Dr. Emilia Abdul Malik. Are you with us now, Dr. Emilia?
Yeah, thank you. Can you hear me? Okay,
I can hear you clearly.
all right.
Okay. Could you please speak?
You cannot? All right, I think I'll change mine.
Okay. It's clearly here.
No,
How are you?
I'm fine, thank you.
Good. So the second keynote speaker is Dr. Fabian. Hello, Dr. Fabian.
Hello,
How
good
are
morning.
you?
Very fine, thank you.
Okay. So before we start our plenary sessions, pardon me, I would like to introduce you to our participants. For the first of our keynote speaker is Dr. Emilia Abdul Malik. So she is a lecturer at University Putra Malaysia, UPM from Malaysia. So she graduated from Oxford University in synthetic organic chemistry. So she is she does research in synthetic organic chemistry, and now teaching and working in University Putra Malaysia. Today's topic that she will talk with us today is modification of zeolitic imidazolate framework with cancer homing peptide for targeted delivery of chemotherapy drug. So this topic is about how to modify the surface of zeolitic imidazolate framework using RGD, arginine glycine aspartic acid. Okay, to shorten the time is yours.
Terima kasih, Madam Pengerusi Majlis. First of all, I'd like to thank the organizing committee of the conference for inviting me to talk today. So for today, I'm going to talk on modification of zeolitic imidazolate framework, or ZIF, with cancer homing peptide for targeted delivery of chemotherapy drug. Okay, I'm Dr. Emilia Abdul Malik from Department of Chemistry, Faculty of Science, Universiti Putra, Malaysia. So I'm sure everyone here knows already what cancer is. Okay, so let me give a brief definition, which from National Cancer Institute, which define cancer as disease in which abnormal cells divide without control and can invade nearby tissue. And usually, chemotherapy, which is a treatment that uses drug to stop the growth of cancer cell, either by killing the cell or by stopping them from dividing, are used a lot to treat this cancer. And although the chemotherapy is very effective, but it comes with a lot of side effects, such as mouth sores, nausea, hair loss. And this is due to killing or inhibition of rapidly dividing normal cell, as well as the cancer cell. And such this kind of normal cell that usually kill as well will be the hair follicle that result in the hair loss. So the chemotherapy is usually non-selective and non-targeting. So in order to avoid this chemotherapy side effect, we propose a targeted delivery approach, whereby we use cancer homing peptide, the RGD peptide, made up of 3 amino acids, arginine, glycine aspartic acid, as what we call as a GPS. It's GPS that would deliver the cancer drug right to the intended recipient, which means to the cancer cell and not to the normal cell. So that would avoid the unwanted side effect. And how does this RGD work? Why is it possible for RGD to home into the cancer cell? That is because this RGD peptide recognize the integrins alpha V beta 3 and alpha V beta 5 that existed on our cell. And these integrins are overexpressed on the cancer cell. That's why we can differentiate between normal cell against cancer cells. And because RGD peptide can recognize this integrin, it can help homing in the drug delivery vehicle that we've chosen towards the cancer cell, and less would then be gone into the normal cell. And the delivery vehicle that we chose for this work is ZIF, zeolitic imidazolate framework. There were 2 that we chose, which is ZIF-8 and ZIF-90. I'll talk about it later. Which this delivery vehicle are porous, okay, in nano size, which are suitable for drug loading. At the same time, it also has high aqueous stability, means that it can be it will be stable in our in our bloodstream. And at the same time, is also stable at a high pH, alkaline pH, but it tend to disintegrate at acidic pH. And this is good for us because the cancer cell has acidic pH, means that it would disintegrate around the cancer cell. Even though it may be delivered to the normal cell, it will be less likely to disintegrate, so less likely for the drug to be released around the normal cell. And at the same time, they are biocompatible. Okay, so the solution for the non-selectivity of the cancer drug will be to encapsulate the drug in stable ZIF and then modify the ZIF with RGD peptides to get to the targeted delivery. And then this will increase the specificity, hence lower dosage will be used, and hence also less side effect will be there will be less side effect. So justification of this work will be that the high aqueous stability of the ZIF will ensure drug release at the target site, and recognition of RGD peptide by integrin will ensure more loaded ZIF reach the cancer cell, and then lower pH surrounding cancer cell would lead to disintegration of the loaded ZIF at the target sites. So in our drug delivery vehicle, we have a porous material whereby we load the drug. Okay, the red dot is the drug, and then on the outside we'll functionalize it with the RGD peptide, where the RGD peptide would then recognize the integrin and bring the vehicle near the cancer cell, and then once it's come near the cancer cell, it would disintegrate and the drug would be released. Okay, so the ZIF itself, again, the zeolitic immunosorbent framework that we chose as the delivery vehicle is actually part of the metal organic framework or MOF family. And MOF is, if you're not familiar with MOF, it's actually an inorganic metal made up of inorganic metal cluster joined by organic linker. So it has inorganic part joined by organic part. So what you have is a metal unit made out of various metal, but for ZIF that we chose here is a zinc. And then we have the organic linker, which is imidazolic linker, we join those inorganic unit together. So there will be they have what we call a join, what you call a very big cluster, a big network, okay, with holes inside for the drugs to be put in. So for the zeolite, for the ZIF, it has zeolite-like structure and have better aqueous stability than other MOF. And the ZIF90 differ from ZIF8 due to the linker, whereby for the ZIF90, the linker is imidazolic carbaldehyde, so it has an aldehyde group on the outside that can allow for formation of imine bond to RGD peptide. So we can make a covalent bond between the ZIF to RGD peptide. Whereas for the ZIF8, it made the linker is 2-methyl-imidazole, so there will be no covalent link between the ZIF to the RGD peptide, but the RGD peptide would what you call make a connective bond to the zinc and held onto the network. Okay, so this diagram showed for the ZIF8 system, where in terms of the synthesis, we put together the zinc metal ion, the solution of zinc metal ion together with the linker and the drug. Okay, for the ZIF8 system, we use gemcitabine, a cancer drug, and we do in situ, okay, encapsulation. So during the synthesis of the ZIF, the drug will be together and the drug will be entrapped within the pores of the ZIF as the ZIF is assembled. Okay, and then the RGD peptide, once the ZIF with encapsulated, the gemcitabine encapsulated ZIF was formed, we then added RGD peptide for it to coordinate to the zinc, and that would form this framework with this gemcitabine inside the pores and the RGD peptide on the outside as the GPS signaling. Okay, so then this the whole network delivery vehicle would then be able to be targeted towards the cancer cell. So it's going to be attracted to the integrin on the cancer cell, and upon reaching that cell, there will be cellular uptake. Okay, and then it's going to disintegrate and release the drug. And that drug would then induce cytotoxicity and cell death towards the cellular apoptosis. Okay, so for the synthesis and characterization of the first system, RGD at gemcitabine and nano ZIF8, as I said earlier, we did one pot in situ synthesis, okay, to encapsulate the gemcitabine, and the loading amount of gemcitabine in the system is 8.6 milligram per gram, about 7.9%. And the encapsulation of gemcitabine in ZIF8 did not alter the crystallinity of the framework of the ZIF8. And if you look here at the PXRD, the peak of the ZIF8, the gemcitabine ZIF8 and gemcitabine RGD ZIF8 all match the simulated PXRD. So the structure of the ZIF8 is robust and able to withstand the condition needed to modify its surface with RGD peptide. And for the characterization of the size of the nanoparticle, the size is found to be within the nano range, and the size increase as we put on the RGD peptide on the outside. So it goes from 76.7 nanometer to 97.9 nanometer. So before putting on the RGD peptide on the outside, you can see the octahedral structure of the ZIF8, but once we put on the peptide on the outside, it loses that kind of octahedral structure a bit. And we use FTIR to confirm the formation of or what you call coordination of the RGD peptide to the ZIF8, whereby we found new signal at 1738 and 1216, okay, responding to the C double bond O and CO bond of the peptide. Okay, and this confirmed the coordination of the RGD terminal carboxyl group within the ZIF8. For the ZIF90 system, okay, we use a little bit different method in terms of the synthesis. Okay, so for this system, we put on cisplatin drug. Okay, so a different cancer drug. So cisplatin is a very traditional, conventional drug used for lung cancer, and still used, although it's kind of earlier drugs for cancer treatment. Okay, and for this we use PVP, polypyridine, vinyl pyridine, as the size controller or modifier, okay, to afford to the nano size of the nanoparticle. And then the two solution, okay, the solution A and solution B were then sonicated to form the encapsulated ZIF within the ZIF90. And similarly, we then modified the ZIF90 particle with RGD peptide. The only difference is that because the presence of the aldehyde group, we can make covalent bond between RGD peptide to the imidazolate linker. So it has a better stability in terms of bonding of RGD to the delivery vehicle. Okay, so for the characterization in terms of the formation of the ZIF8, we use PXRD, and then it confirmed that match with the simulated ZIF8. Okay, so for the reticular material, okay, MOF or ZIF, usually what confirm the formation will be peak below 10 theta. Okay, that is confirming the very ordered material. Okay, very crystalline and ordered material. And then FTIR showed again presence of the RGD peptide once we put on the RGD onto the ZIF90 particle. On top of that, we also confirm using NMR for the formation of the covalent bond. Okay, you see here the formation of the C double bond and the imine bond between the imidazolate linker to the RGD peptide. So for this, we use a solid state carbon-13 CP mass spectrum to show formation of covalent bond between the RGD and ZIF90. At the same time, we also use size exclusion chromatography to confirm presence of RGD peptide to show that we have the RGD peptide within the nanoparticle system. And the pH responsive behavior of the delivery system will confirm when we do drug release at various pH. Okay, so you see here at pH 5, where it will be similar to the cancer environment, we have almost maximum release after 10 hours. But at pH 7.4, similar to the blood, okay, the normal cell, there'll be very little release of the drug, okay, and you get a sustained release when pH is about 6.0. So this is confirming better release at lower pH, nearer or closer to the acidic environment of the cancer cell. At the same time, for the stability test at various pH, we also did that, and again confirm that it's stable at high pH or alkaline pH at 7.4. It's stable, so here it maintained the stability, and still see the peak at about 70 theta, whereas when we immerse it in a very acidic pH for 24 hours, the integrity of the framework now is lost. So you don't see anymore this peak. Okay. So disintegration of ZIF90 occur at acidic pH with optimum release obtained at pH 5, whereas it's stable at pH 7, where I see minimal release during transport or normal cell. So we expect minimum release during transport or at normal cell. Okay, that will also reduce the side effect. And we did also cellular uptake in cancerous cell. So we compare between ZIF8 and RGD with ZIF8 to see how they are uptake in the cell. Okay, so for the untreated cell, we don't see any fluorescent tagging. Okay, so the ZIF8 and RGD ZIF8 were loaded with fluorescein, which is a fluorescent tagging. Okay, and then with the untreated, we don't see any of those fluorescent tagging. But with the ZIF8, you only see some fluorescent tagging, means that there will be some uptake, cellular uptake. Okay, but with the RGD peptide, the cellular uptake is more. We can see more fluorescent tagging. So it's it confirmed our hypothesis that RGD peptide would lead to more cellular uptake for the cancer cell. And we confirm the selectivity of over cancer, selectivity towards cancer cell over normal cell by doing the in vitro cell analysis. So we did an MTT assay, and we noticed that the toxicity of the RGD at gemcitabine nanoZIF8 in the cancer cell, which is A549 cell, is concentration dependent. You see the same trend comparing between 24 hours exposure to and 48 hours exposure. Okay, and if we compare between the cancer cell, which is the bone, and the normal cell, which is the one that is patterned, okay, you can see that it's less toxic towards the normal cell. And the encapsulated particle, okay, encapsulated gem particle without RGD peptide is less toxic compared to the one with the RGD peptide. So this also proves our hypothesis that RGD peptide would improve delivery to the cancer cell, hence would has increased toxicity towards cancer cell over normal cell. Similar observation were obtained for the ZIF90 system. We can see that the RGD modified, okay, encapsulated cisplatin particle were more toxic towards the A549 cell, okay, compared to the normal MRC5 cell. Okay, so the RGD modified particle will be more toxic than just a ZIF90 particle, and the encapsulated cisplatin nanoparticle without the RGD peptide. And we also calculate the selectivity index, okay, of the two systems. Okay, so basically we see better IC50, okay, with the IC50 for the better IC50 for the cancer cell 11.2 against the normal cell at 27, which lead to a selectivity index above 2. Okay, and then at 48 hours, the value goes even higher. So when you have selectivity index above 2, means that it's significant and selective towards cancer cell. Similarly, the same observation is seen for the ZIF90 system. Okay, so comparing between system with RGD, with RGD and without RGD, there will be less different on the normal cell, but a marked different on the cancer cell, okay, which lead to a higher selectivity index in this case. So I suppose if we compare between ZIF8 and ZIF90, whereby ZIF90 has a covalent bond to the RGD, means that it may have a higher RGD attached to the nanoparticle, lead to a better selectivity index for the cancer cell. So as a conclusion, I can say that ZIF8 and ZIF90 was successfully loaded with chemotherapeutic drug and modified with RGD peptide to enhance the selectivity towards cancer cell over normal cell. And then surface functionalization of ZIF8 and ZIF90 with RGD peptide showed enhanced cytotoxicity towards cancer cell. And then that selectivity index showed that the constructed nanostructure is more selective towards cancer cell than normal cell. Okay. And for that, I would like to, okay, so these are the papers that you can refer to. You're interested to know more about this work. And I also like to acknowledge some people, okay, Dr. Nurul Marina and Dr. Adamu Abu Bakar for the work, for carrying out the work. Okay, Professor Dr. Muhammad Basharuddin Abdul Rahman and also Professor Datin Dr. Sharida Fakhrazi and Mr. Kyle Cordova for their collaborative effort, and also University Putra Malaysia for grant Putra GPIPB 2017, grant number 9580901 for the funding. Okay, for that, I thank you all for listening. And before I finish, I would just like to promote, sorry about this, our Master of Coursework program on analytical chemistry, which you can finish in 1 year to get a master degree. So please feel free to look over on our website if you want to know more. Okay, with that, thank you all for your kind attention. Thank you again for the invitation.
Okay, thank you very much, Dr. Emilia. Such a nice and interesting topic and presentations. I think there are many participants who would like to ask you, but we have to save the questions. We will step forward to our second keynote speaker, Dr. Fabian. Hello, Doctor.
Hello.
Hi. So I think before someone's word, so you have a friend who wants to say hello to you, but before it, I would like to introduce yourself to our participant. So, okay. So, Dr. Fabian is Deputy Head of Center for Taxonomy and Morphology at LIB in Germany. So, he is an expert in diversity and evolution of the sailfin silver sites, and his research interest is ecology and evolution. Also taxonomy and biodiversity. So for today's topic, he would like to talk about freshwater fishes in Sulawesi, understanding diversity and biology to enable informed management. So the topic's really sweet with us because we stay in Sulawesi. So Dr. Fabian, if you don't mind, your friend Dr. Anna would like to say hello to you for 1 minute. The time is yours.
Hello, I do not hear anything.
Hello, Fabian.
Hello, Anna. Nice to see you.
Nice to see you online. Good morning. How are you?
Oh, I'm very fine. It's very early in the morning in dark, cold Germany. So I'm jealous that we're sitting in
It's
the
still early.
wonderful hotel.
Okay. I want to say thank you very much for being a speaker today in our seminar. We are really appreciate it, and I hope we can go to the field again in the next time. Okay, thank you, Fabian.
Thank you, Anna. It's a pleasure being here. Thanks a lot.
Okay. So, such a pleasure to have you here, Dr. Fabian. So to shorten the time is yours.
Thank you so much. Let me quickly try to share the screen. Just a second. So there we are. And I do hope that you can see my slides by now. Does it work?
Yes, we clearly see the slide.
Thank you. Thank you very much. So thanks a lot to the organizing team, and thanks a lot for being invited to present the research of our team here today. And at first, big and many thanks to our partners in Tadulako University. Without you, all this would not be possible. And so it's my pleasure to speak as a zoologist about a very different topic compared to the talk we just heard on cancer research, which is super interesting. However, let's now switch to Sulawesi fishes, and we try to understand diversity and biology of the fishes in your wonderful island, also to contribute a bit to what we think should be informed management. So, yeah, that's the first content slide. So I brought a slide of Sulawesi Island first. So usually I always have to tell where the island is. In here, not a big advantage. And you, of course, do know probably all better than I do about the fascinating environment, including lots of fresh water. So Sulawesi is holding wonderful ancient lakes, streams of all kind, rivers. But what about the fish diversity? So I'm a zoologist, specialist in fishes. So I'm puzzled about biodiversity. And in 2011, Lynn Parenti summed up 76 species of freshwater fishes that are confined to freshwater in Sulawesi. And in 2015, Willy Miesen in our group wrapped up the actual literature, came to nearly 90 species, but he found records of more than 200 if you include all the fishes that enter the freshwater. So there's actually much more. And we conclude that there's basic work required. So zoologists need to go to the field to find out what's out there. And also the question is very open. How do those ecosystems work, and what's about the human impact? Well, let's first focus on the lakes. I mentioned the large lakes of Sulawesi. You all know the names, but let's mention them again. So right-hand side, the Malili Lakes, consisting of 3 main lakes, Matano, Mahalona, and Turuti, and small satellite lakes. And left-hand side, Lake Poso, the big one not too far from Palu. And if we start first having a brief look at the Malili Lakes, we find that they are full of endemic animals. And you're all aware in there that Anna is working on shrimps, not exactly from that lake, but from another lake with Focus. And in here, I brought pictures of shrimps that are to be found only in the Malili lakes, colorful small creatures, and also large crabs that are endemic to the area, and snails and mussels and many more animals. So this is really a center of endemism, something very, very special you're having in Sulawesi. In here is a model of the depth distribution of Matano Lake. You're all aware that this is a big lake, but look how deep it is. It's nearly 600 meters deep, so it's among the deepest lakes in the world, and it has the shape of a bathtub. And if you think of that, the age of this lake is super high, so we are talking around 1 million years, it's clear that evolutionary processes probably contribute to this endemic diversity in this single lake. So I mentioned that I'm a fish person. So I had a look at the fish diversity of the Malili lakes over years, and all these small pictures show species that are endemic, so only occur in the Malili lakes area, and some are confined to parts of the lake system. So these fishes in here, for example, only occur in this lake, and there are more species. So we ask what might explain this endemic diversity and how to manage it. Well, if you're interested in evolutionary processes, it makes sense to ask if certain structures of an organism, so its properties, are useful for its ecology. So if there's adaptation to certain modes of resource use, as the evolutionary biologists say. So in other words, do they have special structures like the mouth shape enabling them to feed on certain items like shrimps or insects and so. And the theory says that the structures in the species should differ according to their ecological niche. And this is what we tested already in early studies. So we investigated in detail parts of the morphology, so parts of the bones of these fishes, and related them to what they actually eat in the lake. And so this is a network displaying some groups of feeding modes in these fishes, and we tried to understand if the morphology meets the ecology. And interestingly, it does. So we continued that in depth, and this means really in depth. So we went diving in the lake and collected data describing the habitat. So we asked, does this species use a certain kind of habitat compared to this species, to this, and so on and so forth? This is the outer part of the circle, and the inner part of the circle shows what they predominantly eat. So for example, we have a fish in here, endemic to Matano Lake, that occurs predominantly on soft substrate like sand and mud, and it eats fish egg. Fish egg? Okay. Fish egg comes from other closely related fishes, these sailfin silversides. So this guy here eats the eggs of the other fishes of the Matano Lake radiation. And so you can tell for every species what's its ecological niche. And then you can use genetic methods and try to understand how these fishes are related. And I just sum this up. So we find that the fishes endemic to this lake are super closely related and obviously evolved in that lake. And if we ask how does it work that species evolve in a lake, we can work with something we call fitness landscapes. That means the expectation is with certain structures, like a certain mouth structure, these fishes are able to, for example, feed on shrimp. And this shrimp feeding enables them to harvest enough energy that this basically pays off. So this is called the process of adaptive radiation. And yeah, so we analyzed details of these radiations over a long time, and many people contributed. And I think we learned quite a bit of about the details, how this works. So including the age of these radiations. So the Malili Lake's Selfin silverside radiation is about 2 million years old, which is a bit for a lake radiation, right? And we also found evidence that speciation may proceed with gene flow, which is somehow interesting from an evolutionary point of view. So these fishes evolved in direct contact with each other, which is, yeah, can be summarized as sympatric speciation. And there are many more things to find out. So it's a fascinating lake in the central highlands of Sulawesi. Well, there are also severe problems, especially in this lake. And I'm a fish biologist, and if I see a fish in such a lake, and I know that this fish species does not belong in there, I try to understand, okay, how does it come and what's the potential impact? And one of the species I found is this cichlid species. It's a flowerhorn or lohan. And this is in fact not a natural animal, it's an artificial hybrid. So these fishes are produced for the aquarium trade. And this is a hybrid among different species of fish from South America. And as long as you keep these fishes in the aquarium, it's of course not a problem. It's nice if people have pleasure when watching fishes in the aquarium. But someone somehow put them in the lake. And what happened was that these fishes spread. And I'll show you in a minute some details. And you see the big fish here. And if you look closer, there are numerous small brown dots, and all these are juveniles. So there are several hundred juveniles of that fish. And that's a picture taken underwater in Lake Matano. And as you can see from the plastic debris, it was not far from town area. Well, the cichlid was not the only foreign or exotic species that we found in Matano Lake. So, and we summed our records up. So each colorful square in here means the record of a non-native species. That means a fish that does not belong to Sulawesi, in most cases even not to Indonesia, like this South American suckermouth catfish. And when we come back to the cichlid species I introduced you to, we recorded that species first here in the area of Sorako town, which is a big mining town at the shores of Matano Lake. And then we traced the distribution of that species around the lake over time. And you can see it dispersed rapidly all around the lake. And now it's present basically everywhere. And you might now ask, okay, is it a problem that a South American hybrid fish invaded our lake? And we studied what they feed on. And I can tell you, yes, it's a problem. It's a severe problem. Left-hand side is what juveniles eat. So this appears somehow okay. The majority are aquatic insects. However, if they grow larger, they eat basically everything. They can get into the mouth and they get large, so to that size. So they eat all the endemic animals, like the snails, the bivalves, the crabs, the shrimps, the fishes, and so on and so forth. And yes, this is obviously a severe ecological problem. And when I recently visited the lake, I just can say that there are super clear indications that the ecosystem changes. So the invertebrates, all the small animals, most of that has disappeared from the shallows. And this will, of course, change the ecosystem, or it already changed the ecosystem of the lake. And it might contribute that a wonderful, pristine, clear water, a major freshwater resource in Sulawesi, changes into a less clear, less usable environment dominated by exotic species. By the way, Fadli Tantu, I don't know if he's around. He is quite active in that area, and he even organized contests for catching these fishes and for testing recipes. And this is, of course, super valuable. However, I think there's virtually no chance to really remove it from the lake again. And unfortunately, most people do not at least prefer it as a food. Yeah, let's now have a look at the second large lake, Poso. This is a depth distribution map of the lake, and you can see again something very deep, several hundred meters, again a bathtub shape. And in that lake, there's again a radiation of freshwater fishes endemic to the lake, means only occurring in here, and this time rice fishes, another group of fishes. I'll come to that a bit later in detail. Two genera, Adrianichthys on the right-hand side, rather large species, probably around half of the diversity already extinct. And on the left-hand side, Orythias, small fish species. And I'll show you in a minute why these are quite relevant. So, as I said, we need to find out what's out there first before we can really understand the biodiversity. And this is a snapshot left-hand side of Eco and Jan Möhring, our PhD student, this year at Poso Lake. And there's an illustration of a cable-bound submarine equipped with video camera for exploring the deeper areas, because we can, of course, not easily dive to 70 meters depth or so. Right-hand side, some snapshots. And we, of course, observed animals in the depth. And however, you can also see the colorful fish, the blue one, which is again non-native. It's a cichlid fish from Africa. And Jan analyzed the ecology of the rice fishes again to understand evolutionary background of endemic species. And here's a plot of ecological data relating habitat to species. For example, you see such a vector entitled sand and a vector entitled slope and large rocks. So when collecting ecological data, you can tell which habitat is relevant for which species. And these rice fishes use very different habitats in the lake. So actually, we're focusing on a phenomenon called introgression. That means that this radiation is in genetic contact with the river population. So Poso Lake is drained by Poso River, and this is also inhabited by fishes, and there's genetic contact, and we try to understand the evolutionary background of that, and this is also relevant when talking about management and conservation. So again, in Poso Lake, we found a super large selection of exotic fishes. And this is not all new. So the introductions, the first date back more than 100 years ago. And local people even think that the fishes belong to the lake, but they don't. For example, tilapia. So many people in Sulawesi think tilapia is native. It's not. It's coming from Africa. The same is ikan gabus, the chana. It's not native to Sulawesi, and so on and so forth. So we listed the species and the records to provide some scientific background. And what we found also was this African cichlid. It dispersed super rapidly throughout the lake. Male is blue, female golden yellow, a really beautiful fish, but it doesn't belong there. It comes from Lake Malawi in East Africa. And now it's present in millions in Poso Lake. And we again looked at the impact and can conclude they feed again on nearly all of the wonderful native animals that are actually your natural heritage in central Sulawesi. Well, these are big ecological problems, and they will turn into economical problems because lake ecosystems change and people cannot continue using them in the way they used to do. I promised that I will introduce you a bit into the question why we are interested in rice fishes. This is basic research. Right-hand side, you see a snapshot of our rice fish team around Julia Schwarzer, who also contributed a lot to this research in here. And it all started with a nice discovery. So more than a decade ago, I described with two colleagues, Reni Hadiati, who unfortunately passed away by now, from former LIPI in Bogor, and Arne Nolte from Plön. At that time, we described a rice fish species, a small fish, carrying the eggs. It's endemic to a single spring pond in Rantepao, Toraja area. And this is somehow super fascinating. And this whole team formed to understand how this reproductive morphology evolved. So super interesting from a basic research point of view. This is such a female rice fish, and she carried these eggs. You can already see the eye of the embryo, and these eggs were attached to the female by filaments. I go back one slide. So these are the eggs attached to the female, and when you remove those eggs, you can only see those filaments. And there's even the hypothesis that the mother fish provides nutrients to the eggs. And this was among the questions we wanted to ask using these small rice fishes. And so a lot of work was done, for example, by Leon Hilgers, who's now in Frankfurt, who explored in detail the transcriptomic background of the tissues related to this brooding morphology, and by Alina Schüller on the right-hand side, who analyzed in detail the histology of this brooding morphology. And the story is super complex, but the take-home message is that there's a novel tissue that evolved in that fish, and it's explained by inflammation processes, which is somehow weird. But this reproduction morphology induces inflammation processes in the fish, and this inflammation obviously leads to the formation of a special tissue that then contributes to enabling the fish to hold these eggs. So super, super strange adaptations to be found only in a single spring pond in Rantepao. Well, you can also continue looking at details of the structure of the fish. This is a rice fish in a tomographic unit in our lab, enabling three-dimensional models. And you see again the rice fish. And so this looks like an X-ray of the fish. And here in purple, you see a tiny, tiny bone. the pelvic girdle, and this bone holds the fin, the big fin, that again holds the eggs when these fish carry the clutch of eggs. And Toby Spanke from our team, he investigated the role of that tiny bone and also the structures of the skeleton of the ribs, asking how does this pelvic brooding morphology actually work? And one conclusion is that you can look at any detail of these fishes and you will find that the morphology reflects the ecological trait these fishes need for their reproduction. So super, super fine scaled morphological adaptations. Well, coming from tiny, tiny structures to the large picture again. So I said that we also worked on streams of Sulawesi, and on that map in here, you can see some color codes showing geological entities of Sulawesi Island. I will not focus on geology now, we'll go more into ecology, but what you can see in here that there are numerous small rivers and lakes, and we asked what determines species diversity in the streamfish communities and how can all these species coexist? How does it work? Yeah, and as soon as the next slide loads, yes, you can see our map including sampling sites across Sulawesi. So this was a major enterprise in 2019 with colleagues from Manado University, and the analysis of this part of the projects are predominantly done by Leta Vantania, who's now finishing her PhD with me here in Bonn, coming from Unsrat. And our team went along long journeys along the island and investigated streams from lowland to upland to highland, with a focus on coastal drainages, so not the big rivers, not the lakes. You can see some pictures of wonderful field work in here. We worked a lot with electrofishing, which is quite efficient for recording freshwater fish diversity. And you can also imagine that this is somehow intensive field work, and this is how it looks like if fish people are successful in catching animals. We do not only have to determine and measure, but also to record environmental parameters of these fishes. And this is Leta here with Willy Miesen, who joined that trip. And what we did in here was to conduct point abundance sampling. That means that you select a stream site and run a zigzag course along that stream and record the presence of fishes every few meters and relate these to the environment. So we record depth, wood, elevation, pH, oxygen, and so on and so forth. And over the time, with increasing number of sampling points, of course, the number of species recorded increases. And so we see that we somehow reach a saturation. So we somehow capture at least a fair proportion of the diversity. And these are some snapshots of what's dominant in the Sulawesi stream communities, like these gobies, with a emphasis on climbing gobies, the colorful ones, and the sleeper gobies, the eleutrids. But there's much higher diversity from freshwater moray to Rheacichthys to guppy and so on and so forth. So also introduced species in here. Most of the species were native that we recorded. However, also in the stream, a fair proportion of exotic species. And on the right-hand side, you can see that the majority of the species is classified as peripheral. That means these are species that have, at least in their life cycle, at some point marine tolerance. So the species require somehow access to the sea. And if you now analyze the ecological properties of the species, we can see that elevation is the most relevant vector. However, also water chemistry like pH and conductivity and the substrate plays a role. So going more into detail, you can now analyze the ecological niches species by species, for example, along the elevation gradient, and tell what's most important for the fish communities, ranging from species that are typical for brackish water to highland ones. And I quickly just go through it, and the message is that fish species use virtually all aspects of the stream ecosystem. So you can see in here, they use basically the whole ecological space available. Well, substrates matter. They also change along altitudes, of course. And yeah, the current velocity, of course, also matters for stream fishes. Summing it up, elevation and water chemistry are of strongest relevance, and the microhabitat segregates among the species. And what's next, this is also on Leta's to-do list for finishing her PhD, is to check in detail the morphology of the species recorded and relate them to their ecological niches. So these are some tomographic models, and Leta produced nearly 130 of those so far. And we will learn much more about the ecomorphology of Sulawesi stream fishes soon. Yeah, I will summarize a bit on the threats, because this is quite relevant for management. So I think I've shown you that invasive exotic species are a severe problem in Sulawesi. Also, eutrophication is a real issue in lakes and streams. Just remember, you would like to see some clear waters in water reservoirs, not greenish brown algae communities, right? We do, of course, see severe habitat destructions coming with developmental steps in certain areas, forest destruction, and especially also in the Sorako area, the gigantic surface mining activities that just remove complete ecosystems and affect lakes. And of course, hydropower energy appears to be green, but from a fish point of view, it's a major issue because it blocks the access of fishes to the sea. And you've seen that in Sulawesi, nearly everything in the streams depends on access to the sea. So if you block a river completely, you just change the ecosystem completely, and local people will realize, because the whole ecosystem will change. So this is an illustration here from Europe. We are, of course, facing the exactly same problems like the habitat change. This is an illustration of the natural river providing a lot of habitat and biodiversity. And this is what we are having right now in many areas of the European Union, channels which look beautiful but are ecological disaster. And nowadays we are going back to break up the channels to restore the ecosystems, but it's a super, super high effort. Well, that's it. My time is over. I would like to thank our team, and this illustration does not display all people that contributed, only the people that contributed to the slides I've shown in here. My severe thanks to the whole team of colleagues, and thank you again for having me here and listening to Fish Talks. I'm very happy to receive questions. Thank you.
Okay, thank you very much, Dr. Fabian. It's such a nice and really interesting topic. So I think we have maybe 10 minutes for question and answer. So if there is anyone want to ask to our keynote speaker, Dr. Emilia or Dr. Fabian. Is there any questions from participant in this room? How about in the online participant? Is there any questions? Please raise your hand if you have any questions to both of our keynote speaker. Is there any questions? Okay.
Oh. Okay. Okay. Good afternoon, Dr. Fabian. My name is Mustafid Rashid from Tadulako University. I ask you about you say invasive species can reduce native species population. So how we can control or reduce population of invasive species, especially fish in Sulawesi. Thank you.
Thank you very
Okay,
much.
thank
This
you
is a
very much.
super good question. So actually,
Yes, yes.
actually really removing an exotic species from an open ecosystem, I don't want to say it's impossible, but my belief it's impossible. The challenge is super high. So people tried that in other parts of the world, like with invasive crayfishes in freshwater streams in UK, and they tried to poison whole drainages to remove invasive species. And in the end, they were not successful. And so you, of course, damage the ecosystem if you use anything like that severely. So I would say it in another way. It's time to wake up, to be aware, not to introduce more, because you never know if the next species that arrives will be another full hit, like the cichlid species from East Africa or the hybrid cichlid. So some of the invasive or some of the exotic species do not really provide a major problem. However, you cannot predict, and this is a bet. So I think the first thing is be aware, do not add foreign species. So you have a wonderful biodiversity in there, and it's your natural heritage. Putting in species from other areas of the world just, has the potential to introduce disasters. However, I do not have hope that we are able to remove any of the invasive species from the lakes. I do not have that hope, unfortunately. I see the super severe consequences for the ecosystems. In the rivers, they're more open than lakes. It's probably even worse. So the first step would be to raise awareness that stocking of non-native species is not a good idea at all. This will be one, or this is already one of the major environmental issues, especially in Southeast Asia.
Okay, thank you. Is there any questions? Please raise your hand if you have any question. No questions. So I have questions to Dr. Emilia. So you synthesize zeolitic or ZIF8 and ZIF90. For the ZIF8 encapsulated and into a gene, and the ZIF90 is combined with a cisplatin. I mean, what is the difference? What is the specifications that the ZIF known as ZIF8, and for the cis, it's known as a ZIF90.
Let me just rephrase the question. Were you asking whether I have a combination drug in one of the system in combining gemcitabine and cisplatin in one system?
Yeah.
Was that the question?
What is the difference for the 8 and 90? There's any specification for it?
Okay, in terms of the size, they are the same. So they basically the spacing between the zinc, between the two zinc is the same because it's the same linker. The only difference is that with the ZIF8, there are no covalent bond between the ZIF to the RGD peptide, whereas for the ZIF90, there are covalent bond between the RGD peptide and the framework. So when we start the work, we were using ZIF8, and we were worried that we're not going to have a good bond between the ZIF to the RGD peptide. So if we don't have RGD peptide surrounding the ZIF, then we were not going to see the selectivity, because then we wouldn't have the RGD peptide helping us homing towards the cancer cell. So with the ZIF90, it's more clear-cut because we can make the covalent bond with RGD peptide, so we can guarantee that we have RGD peptide on the surface of the framework. So based on that, our hypothesis is a bit more strong in terms that the RGD peptide will be able to carry our drug to the cancer cell. But in terms of the double drug or dual drug combination, we have not done that yet. But I suppose gemcitabine and cisplatin going to be a very good combination because they have different activity. How they react with the cancer cell is different. So this is something that we are going to work on in the future, I suppose. Yeah.
Okay, thank you. This is my last questions for you, doctor, because this is really interesting. How about the interactions between the RGD with the ZIF? I mean, does the RGD covalently bond with the cis or the ZIF?
That's why I was just saying just now. With the ZIF90, there is a covalent bond. So we are making imine bond between the aldehyde functionality on the imidazole linker to the RGD peptide. So there's covalent bond. But with the ZIF8, there are no covalent bond, but we expect because we can confirm that there are RGD on the nanoparticle. So we assume that the RGD peptide having the carboxyl group, they were able to make a coordinative bond to the zinc, so filling up the empty coordinative site on the zinc itself. So there's no covalent bond for the ZIF8, but there are for the ZIF90.
Okay, thank you very much.
Okay, sure.
There-
Thank you.
Is there any questions? No, no questions anymore. My last questions for Dr. Fabian. I would like to know, Doctor, as we know that minerals are exist worldwide. So is there any possibility that the mineral affected the morphology of the fish species?
Oh, yes, of course. So our ecological data clearly show that water chemistry matters for determining the distribution of the fishes. And this includes conductivity, summarizing the mineral load in terms of ions of the water. So we know that fishes differ in the use of the habitat also according to minerals. So, and of course, especially the skeleton requires minerals. And so I would answer the question, yes, the availability of minerals will, of course, determine the presence and distribution of at least parts of the species community. And yes, so of course, minerals matters to fishes, and different species require different amounts of different minerals dissolved in water. So I don't know if this answered your question somehow.
Yeah, I think it's answered my questions because as we know that fish has many color, so maybe the color affected by the mineral content in the water. That's what I think.
Yeah, so color in fishes is a complex thing. So there are different mechanisms producing color in fishes. And this might be due to structures embedded in the skin of the fishes, or this might be due to substances embedded in there, like special kinds of proteins. And so it's not that easy that they can really use a mineral for producing color. It's more complex. But on the other side, it's clear that the environment, including the mineral load of the water, affects the surrounding of, or is the surrounding of a fish, and of course affects the populations. Yes.
Okay,
Thank
thank you very much. Is there any questions? No questions? If there is no questions, then I think we are at the end of the plenary sessions 3. So okay, thank you very much for Dr. Emilia binti Abdul Malik and Dr. Fabian Herder for joining us today in the 5th international seminar on science and technology hosted by Tadulako University, and this is the certificate for both of you. Okay, thank you very much, Dr. Fabian Herder and Dr. Emilia Abdul Malik. Once again, thank you and I hope we can meet in the future.
Thank you again. Terima kasih.
Terima kasih,
Yo,
Prof.
terima kasih.
Terima kasih, thank you.